Hello friends, welcome back to my channel, and today in this video we are going to study about cells of lymphoid lineage. Now in lymphoid lineages we have cells called lymphocytes. These are the principal cells of adaptive immune response. Lymphocytes consist of mainly B cells and T cells. Apart from that we have NK cells also and NKT cells also. 20 to 40% of circulating white blood cells are lymphocytes only, and they constitute 99% of cells of the lymph. Now, lymphocytes consist of B cells, T cells, and natural killer cells. B cells and T cells are distinguished on the basis of surface protein, which are known as cluster of differentiation, CD signature. We call it as a CD signature. In the next slide, we'll see what are these signatures and how we can use the signatures to distinguish among T cells and B cells. Now, B or T cells also expresses an antigen specific receptor on their surface because they have to bind with the antigens. Naive cells means in case of both T and B cells, naive cells we call those cells that are yet to encounter antigens. Now, once they contact with antigen, the naive lymphocytes undergo proliferation and differentiation to form both effector cells and memory cells. Effector cells means that perform function. Memory cell will help in killing the antigen for the future attacks. In terms of this first attack and the future attack, we use the term primary response and secondary response. Primary response means first encounter with the antigen. Secondary response means second encounter with the antigen. So it is basically like that. Whether it is a T or B cell, we call this cell as a naive cells that are yet to encounter the antigen. Once this cell encounter any antigen, they will differentiate into either the effector cells or the memory cells. Effector cell will go and have its effector. So they will attack the antigen, and memory cell will be stored in our body for future attacks with the same antigen. So this is the process. Now, in the last slide, we learned that these T cells, B cells or NK cells, on their surface, they consist some markers. And with the help of these markers only, we can able to distinguish T cells and B cells, and also we can able to know with which cells these cells interact, because they interact on the basis of these markers or receptors only, and we call it as a cluster of differentiation signature or CD signature or designation. Now, CD2, the function is adhesion and signal transduction and present in T cells and NK cells, TH and TC. In case of T cells, we have two main subdivision, that is TH cells and TC cells. TH stands for helper. TC stands for cytotoxic. Cytotoxic means toxic for the cell. So they will kill the cells that got infected with antigen. Helper cell means they will help other cells. They'll help B cells, they'll help macrophages, they will help even the TC cells also. So like this. So helper cells means they help other cells. So the ratio of concentration of TH and TC cell in our body is 2:1. So helper cells are more in our body compared to cytotoxic cell, because this is more important compared to TC. Because this TH cell will help B cell also, macrophages also, TC cells also in their activation and their effect. So TH cell is more important compared to TC cell because it is helping all the cells. So CD2, which helps in adhesion and signal transduction, is found on T cells and NK cells. Similarly, CD3 help in the signal transduction, found only on T cells. CD4 is the marker by which we can identify T cell because it is specific to only TH cell. So we can say if any cell contains CD4 marker, that means that cell is a TH cell. So this is a marker that helps to identify the TH cells, and they bind with class 2 MHC molecules. This will understand what is MHC molecule and what do we mean by class 1 or class 2. Coming to CD5, the function is unknown and present on all the cells. CD8, it is present in on the surface of TC cell. So these two, CD4, only specific to TH cell. So it is the marker that helps us to distinguish the TH cell. CD8, only specific to TC cell. So it's the marker that helps us to distinguish the TC cell. Clear? CD16, found only on NK cell. Similarly, CD19 for B cells only, found only on the surface of B cell. CD21, found only on the surface of B cells. So like this, various CD molecules or signatures are there, which helps us to identify the cells, the B cells, C cells, and NK cells. Also helps us to identify its functions, because each CD signatures performing one particular function. So on the basis of these CD signatures, we can say which cell it is, what function it can perform, and also what other cells it will bind. For these, the CD signatures are important. Now coming to B lymphocytes. For the B lymphocytes, the name B. Why B? From the site of maturation. In case of humans, mice, and many other mammals, the site of maturation of B lymphocyte is bone marrow. And in birds, it is bursa of Fabricius. So both stands with the letter B, hence B lymphocyte. B lymphocyte contains BCR. This is B lymphocyte. It will contain BCR on its surface. BCR is nothing but the antibody molecules, because B cells are responsible for secreting antibodies in our body. Activated B cells differentiate into effector cells, which are known as plasma cells. So this is B cell. We call it as a naive B cells until it will encounter any antigen. Once it will encounter antigen, it will form effector B cell and the memory B cell. The effector B cell will release the antibodies in the blood, and hence effector B cells are known as plasma cells. Memory B cell will contain the antibody bind on its surface for the future attack with the same antigens. Clear? So these plasma cells, these are highly specialized for the secretion of antibody, and they die within 1 to 2 weeks. Coming to T lymphocytes. Why the name T lymphocytes? Because their site of maturation is thymus. T cell also contains receptor like B cell, and this is known as TCR, that is T cell receptor. T cell receptor recognize antigens which are presented by antigen presenting cells. The B cell, because they contain antibody as a receptor, they can recognize antigen without the help of any other cell, and also without, they don't need processing of antigen. But coming to T cells, they can recognize antigen with the help of TCR, but as said, these antigens should be presented by any antigen presenting cells. Now these antigen presenting cells will present the antigen to the T cell. So this receptor of antigen presenting cell, which is holding this antigen and presenting it to T cell, this receptor is known as MHC, that is major histocompatibility complex. So the antigen presenting cell is presenting the antigen to T cells. So they will present the antigen to the T cell by binding with a receptor, and this receptor with which the antigen is bound, we call it as a MHC, that is major histocompatibility complex. Now T cells have two major divisions, TH cells and TC cells. TH cell, as we learned in the last slide, contains CD4 on their surface, and they recognize antigen presented by MHC class 2. If this T cell is TH cell, then this receptor or this MHC is class 2 MHC. If this T cell is TC cell, then this receptor or MHC is class 1 MHC. So this is the difference, that TH cell will recognize the antigen when presented by antigen presenting cell having class 2 MHC, whereas TC cell will recognize the antigen with the help of class 1 MHC. The ratio of CD4 plus CD8, why it is written CD4 plus? Because TH cell contains which marker? CD4. So we can write it as a CD4 plus. Plus means indication of presence. So CD4 plus. TC cell contains CD8. So we can write it as a CD8 plus, indication of presence of CD8. So the ratio is, I already told you, 2:1. Effector CD8 CD8 plus T cells. CD8 plus T cells means we are talking about TC cells. The TC cells are only known as CTL, that is cytotoxic T lymphocyte. TC, cytotoxic T cell only. So, cytotoxic T lymphocyte. The CTL monitor the cells of the body, eliminating any cells that display foreign antigen complex with class 1 MHC, such as virus-infected cells, tumor cells, and the cells of a foreign tissue graft. To proliferate and differentiate optimally, this naive CD8 plus T cell, that means talking about TC cells, also need help from mature CD4. In the last slide only, I told you that CD4 means TH cell. TH cell means it is the helper cell. It will help B cell also, it will help macrophages also, it will help TC cells also. Correct? So, these TC cells will bind with the antigen presented by MHC class 1. Along with that, they also need the help from TH cell. Effector CD4 plus T cell, that means TH. Now, these TH cells have multiple subsets like TH1. They will help to activate TC cell. TH2, they activate mainly the B cells. TH17, they help against fungi and provide cell-mediated immunity. TFH, they helps in the regulation of B cell development. Correct? So T cells, or we can say specifically TH cell, they have many subsets. TH1, TH2, TH17, like this, they have many subsets. So most importantly, 1 activates TC cell, 2 activates B cell, as well as macrophages also. So we have T cells mainly divided into 2 categories, TH and TC cell. TC means it will kill the cell. It will directly kill the cell, whereas TH means helper, it helps other cells. Now TC cell will recognize the antigen when presented by MHC class 1. TH cell will recognize the antigen when presented by MHC class 2. Now, depending upon the antigen, whether the antigen is intracellular, extracellular, is it a bacteria, fungi, or viruses, different subset of TH cell gets activated and helps to affect other immune response like that. See here, antigen presenting cell presenting the antigens to CD4 cell. CD4 means TH cell. Now, they can differentiate into TH1, TH2, TH17, THFH, THREG, that is for regulation. And similarly, they will help to activate other cells. So these are about T lymphocytes. The next is natural killer cells. They do not express any antigen-specific receptors and the part of innate immunity. They contain certain cytotoxic granules and constitute 5 to 10% of lymphocyte. Their function is they attack tumor cells and cells infected with virus. That means their function in our body is to protect us from cancer cells, the cells that became cancer or infected with any virus. Now, like B cells and T cells, they do not have any receptors on their surface. Now, how do they recognize self and non-self then? In case of T cells, we learned that T cells recognize antigen that are presented by antigen presenting cells, but these antigen presenting cells should contain some MHC on their surface. Now, for TH and TC, we learned that TC cell will recognize the antigen when it is present with MHC class 1 and TH cell MHC class 2. Now, MHC class 2 are present on all the antigen presenting cells. But even all the normal cells of our body, they contain this MHC class 1. So, when all the normal cells contain MHC class 1, then if any cell that do not have this marker on its surface will be recognized by NK cell as defective, because all the normal cells of our body contain MHC class 1. So if any cell do not contain MHC class 1, that means that is a defective. So that's how NK cell recognize that the cells that are not normal in our body, that means they can be cancerous or got infected with virus. So that's how they help to identify and then downregulate some tumors or viral infected cells. NK cell also has antibodies on their surface so that they can bind antigens with the help of those antibodies. Now we have one more set that is called NKT cells that share the features of both T lymphocytes and NK cells. So NKT. So they express CD4. We learned in the last slide NK cells don't contain any receptor, but it is a combination of NK and T cells, so they express CD4 also, and like NK cells they have antibody receptors also. So activated NKT cells can release cytotoxic granules that kill target cells, but they can also release large quantities of cytokines that can both enhance and suppress the immune response. These are mainly involved in human asthma and inhibit the development of autoimmunity and cancer. So that's all for this video about cells of lymphoid lineages. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.